Propagation of cannabis. Sexual versus asexual propagation of cannabis. Cannabis can be propagated either sexually or asexually. Seeds are the result of sexual propagation. Because sexual propagation involves the recombination of genetic material from two parents, we expect to observe variation among seedlings and offspring with characteristics differing from those of the parents. Vegetative methods of propagation, cloning, such as cuttage, layerage, or division of roots are asexual and allow exact replication of the parental plant without genetic variation. Asexual propagation, in theory, allows strains to be preserved unchanged through many seasons and hundreds of individuals. When the difference between sexual and asexual propagation is well understood, then the proper method can be chosen for each situation. The unique characteristics of a plant result from the combination of genes and chromosomes present in each cell, collectively known as the genotype of that individual. The expression of a genotype, as influenced by the environment, creates a set of visible characteristics that we collectively term the phenotype. The function of propagation is to preserve special genotypes by choosing the proper technique, to ensure replication of the desired characteristics. If two clones from a pistillate cannabis plant are placed in differing environments, shade and sun for instance, their genotypes will remain identical. However, the clone grown in the shade will grow tall and slender and mature late, while the clone grown in full sun will remain short and bushy and mature much earlier.